Thanks, Matt. I forgot to send a CV or a bio sketch, so I'll introduce myself quickly. My name is Adam Kim. I'm now a research associate in the Nagy lab at the Cleveland Clinic in Ohio. I've been in this lab for about 3 years now, I think. And previously, before this, I actually this is my second postdoc. Before this, I did another postdoc where I looked at malaria. And the goal of my postdoctoral career was to do bioinformatics. So actually my graduate school work was in yeast cell biology. And then I made a concerted effort career-wise to move into bioinformatics, as well as to eventually move into either a disease or some kind of human system, which is what I'm going to tell you about today. So in the Nagy lab, we're primarily interested in looking at alcohol-associated liver disease. So to give you an idea about what happens to you when you drink alcohol, so a normal liver, looks like this, is kind of pinkish red. When you drink, if you were to do a binge of alcohol tonight, if you were to drink a binge of alcohol, you will undergo mild steatosis. So most people do this, about 80-90% of people. And this is characterized by triglyceride accumulation, increases in these liver enzymes, ALT, AST, in the peripheral blood, as well as increased inflammation, some cytokine expression. But then if you were to chronically binge drink or just chronically drink alcohol for a very long period of time, you will eventually, like over the course of your entire lifetime, you'll eventually develop, very likely you'll develop fibrosis, and then, which is the ECM hardening of the liver. And then over the course of time, you will also get more and more damage will accumulate. And then eventually the liver will stop working. And this is what's called cirrhosis, where hepatocytes, everything is starting to fail. And then among a rare population of these people, you can develop hepatocellular carcinoma. In the Nagy lab, we're interested in understanding how throughout this spectrum, the immune system really contributes or progresses disease. So we look at this disease called, and specifically we look at this disease called alcohol-associated hepatitis. The idea here is at any point along this stage, you can get some kind of infection, something else happens that causes massive inflammation into the liver. Immune cells infiltrate. There's a ton of hepatocellular death. And what's interesting about this disease is it has really significant mortality. So about 40% of people who get AH die within 3 months. So we think of this as kind of two ways to die by alcohol. You can be a career drinker and in your 60s develop cirrhosis or hepatocellular carcinoma and die of end-stage liver failure. The alternative way is you could be in your 30s drinking heavily and then suddenly develop an infection in your liver that kills you. So when you think about alcohol liver disease, it's really totally systemic. In addition, when we think of alcohol consumption, we think of a lot of the brain effects, and there are tons of brain effects. We also think of the liver as this major place. So in our idea of alcohol consumption, when you drink alcohol, you actually do get this small gut barrier dysfunction. So bacteria from your gut leak into the liver. And then your liver, being an innate immune organ, has to deal with that mild endotoxemia. So the Kupffer cells and the cells in the liver deal with that, the liver immune system. As well as your peripheral immune system also can come in and infiltrate and deal with the infection. We know that alcohol affects the peripheral immune system. We know alcohol affects the liver directly because the liver has to digest the alcohol. And we also know that alcohol affects your adipose tissue. It greatly affects the muscle, and all of these systems interact and communicate with each other. So for my talk, I want to specifically focus on really the peripheral immune system in the liver, as well as a little bit of how the gut works in all of this. So right now, if you were to binge drink alcohol, so if you were to go up to, in the US, our legal limit is 0.08% our blood alcohol percentage. If you were to drink enough alcohol right now to get that percentage, your blood alcohol would go up, but also your serum endotoxin would also go up. But it's a very minor quantity. So when we think of sepsis, we're thinking in the micrograms of bacterial LPS in your bloodstream. In alcohol, we're talking more on the 100 picogram. This is 70 to 100 picogram per mil endotoxin in your bloodstream, in your peripheral bloodstream, from the consumption of alcohol, from a binge drink of alcohol. And there are slight differences between men and women as a result of basic differences between men and women in alcohol metabolism. All right, and then one other thing about this is alcohol-associated hepatitis patients have just basal increased LPS in their bloodstream. So there's they just, as being a sick patient with AH, they have elevated LPS, and it is once again similar to what I just said, about 70 to 100 picograms per mil. All right, so for my project, I want to look at this gut-liver axis and how the peripheral immune system plays a role. And so here's basically our model. We think what happens is when you consume alcohol, gut barrier dysfunction happens, which leads to the leakage of gut products into the peripheral, into the portal immune system, and then eventually the peripheral circulation. And then LPS from there signals through TLR4 to activate monocytes. And once again, this is very low. We're calling this ultra low dose LPS models. If you were to read the literature of papers that do LPS treatments, it's typically on the order of 10 to 1,000 times more concentrated LPS. So we're doing this very ultra low dose LPS model. And we had basically two questions we wanted to ask. The first was answering this question that has been known about AH for a long time, which is that AH patients are just hypersensitive to LPS. So if you look at TNF as kind of a model for cytokines in an immune response, we see in a typical patient, if you're to treat a typical healthy person with LPS, you would get this mild TNF response, but in AH, we get this exacerbated response. So what is the cause of that was the first question I want to ask. The second question I want to ask was, as the gut leaks products into the bloodstream, we mostly think of it as being a endotoxin problem of bacterial LPS. But what about every single thing else that is also in the gut? What about the viruses, the fungi, potentially other parasites, as well as DAMPs, so damage associated molecular patterns that come from the host. So a lot is known about how alcohol affects the immune system. Here's just some of the things that we've looked at in our lab. So for example, we know alcohol affects complement. We also know alcohol through this gut LPS thing can activate TLR4. And then another lab, as well as us, have looked at how fungal byproducts can activate this gene called dectin-1, and other things can activate this other gene called Minkle. And I'll get more into dectin-1 and Minkle in a little bit. So to give you a little bit more background of the biology of the liver is, this is kind of the closest up picture you can get of the liver, where you have hepatocytes, which are, very metabolic. And then beneath the hepatocytes are this sinusoidal, the sinusoid of the liver, where basically three different liquids mixed together, which is your peripheral blood, your portal blood, as well as your bile, all mixed in the sinusoid. And here we get Kupffer cells, they're dendritic cells, they're also endothelial cells, as well as other NK and NKT cells. During alcohol liver disease and any kind of injured liver, we get activation, we get hepatocyte stress, we get stellate cell activation leading to the fibrogenesis. We also get changes in the endothelial cells, as well as activation of the resident immune cells and infiltration of monocytes, which is very important. So because human liver immune cells are difficult to study in kind of a functional way, especially from patients, we looked for another source of cells that we can study in an easier way. And for our lab, we chose to look at PBMCs for a couple of reasons. First of all, we know the infiltrating monocytes come from the PBMCs. So we thought if we can characterize these cells better and what's going on in the peripheral immune system, we can better understand what's happening in this disease. So because I'm primarily interested in bioinformatics and applying sequencing technologies to understand problems in health, I chose to do single cell RNA-seq of these PBMCs. And what we did is we isolated it from 4 healthy patients, age-matched healthy controls, as well as 4 AH patients, where their end-stage liver disease score was very high. So these are patients with severe AH. And actually, just to add one extra layer of complexity, we took the cells and ex vivo stimulated them with this low-dose LPS that we had mentioned before, this ultra-low-dose LPS of 100 picograms per mil. And we treated them for about 24 hours to kind of replicate the long-term exposure of LPS that a lot of these patients would experience. And then we performed 10x single-cell RNA sequencing. All right, so here's the data in accumulation. So here's everything. This is a clustering map of the different cells. There are B cells, there's a lot of T cells, and then the cell that we're primarily interested in, which are the monocytes. and we observed 4 clusters of monocytes with distinct features. So I want to get into that in a little bit. But before we get into that further, I wanted to say one thing. We did observe what many other people have shown before, which is that in AH, there are just more monocytes. So in the healthy patients, we generally see an even distribution. We see this distribution of cell types. So maybe less than 20% monocytes. B cells are in a tiny proportion. And then most of the cells that we were looking at are, different kinds of T cells. But then in AH, we get this huge increase in monocytes. And here's another way to look at that. As a changing population, we see this huge increase in monocytes. All right, so then what we did was we wanted to look at how genes were differentially expressed between the different populations. And as you can imagine, it's a little bit of a complicated analysis, because we can compare healthy control basal to AH basal and come up with some baseline difference. We can also look at how LPS affected these cells. So here's our second and third comparison, how the healthy control basal respond to LPS and how the AH cells respond to LPS. And then finally, we can look at how the LPS responses were different between the two groups. And the thing I want to point out is a majority of our differences, the greatest number of changes that we saw were in fact in our monocytes, which is not surprising because the monocytes are the TLR expressing gene cells, and they should be the quickest and the fastest response to LPS. But what was interesting to us immediately though was the CD14 monocyte cluster 1 had a ton of changes in all of our comparisons, 1, 2, 3, and 4. But then CD14 cluster 2 had fewer changes in the healthy control as well as in response to LPS. So we wanted to understand that a little better. And this is what we found. If we looked at what genes were differentially expressed in response to LPS between these different clusters, we see something pretty interesting. So let's start with this one. Healthy control CD14 cluster 1 had a huge number of genes upregulated in response to LPS. But these are totally unique to this cluster. If we look at healthy control cluster 2, there are a smaller number of genes differentially expressed, and there's a huge overlap between cluster number 2 in healthy controls and in AH, meaning that this healthy control CD14 cluster 2 and in healthy controls and in AH behave very, very similarly. But what's interesting is the AH CD14 cluster 1 behaved totally differently, had almost no overlap from what they originally would do in healthy controls. And now these cells actually behave more like the cluster 2. So another way to look at that is to look at just a specific cytokine. So here's all the data. I'm going to break this down a little smaller. So if we think about 4 monocyte clusters, what we observe is in response to LPS, only one of these clusters really expressed IL-1 beta or CCL2, a cytokine and a chemokine. There's a mild response in cluster 3 and the CD16 cluster. But what's interesting is in AH, all 4 of these clusters now become IL-1 beta and CCL2 expressing cells in response to LPS. So what we think is happening here is that we're getting a change in how these monocytes behave in AH. Whereas normally, in response to LPS, only one subset of cells would respond with pro-inflammatory cytokines. But in AH, every single monocyte becomes a pro-inflammatory cytokine producing cell. And what we think is happening, and then just to go finer into the genes that we're seeing change, if we look at what genes were upregulated and shared between all 4 of these cell types, we only really see a handful, some chemokines upregulated. But when we look at what's shared between cluster 2 as well as this AH cluster 1 that has now become much more similar to cluster 2, we see a huge overlap in genes upregulated. And these are all your pro-inflammatory cytokines, a lot of these metallothionein genes, as well as some other CD and immune-related genes. And then the question that we had next was, this healthy control CD14 cluster 1, they upregulated something that is no longer being upregulated in AH. So what were these genes? And just to kind of summarize, I don't want to go into this in too much detail, but what we saw was these genes that were no longer expressed in AH are actually interferon-associated genes. So genes related to interferon signaling, antiviral responses, as well as a lot of other genes that we would associate with anti-inflammatory gene responses. So what we think is happening in AH is in a normal healthy control in this low dose LPS, we would get a variety of responses. One being pro-inflammatory genes to deal with the infection, as well as upregulation of genes that would be used to deal with maybe potential viruses coming along or other or the need for resolution of infection later. But in AH, what we're getting is every single monocyte becoming a pro-inflammatory cell. And we think that this explanation, as well as what I told you before, which are there are just more monocytes in AH, explains why we're getting this exacerbation of LPS responses of TNF expression in LPS. All right, so the last thing I want to tell you about pretty quickly for the second half of my talk is this receptor I told you about before called Minkle. So Minkle is a C-type lectin receptor. It is another PRR, pattern recognition receptor, and it's in the lectin family. So the idea is that it senses a larger variety of potential targets, specifically a lot of sugar moieties. So Minkle is famous because it was really one of the first genes that thought to be able to sense tuberculosis. So it senses TDB, which is a tuberculosis core factor. It was interesting to us for a while because it's also thought to sense this DAMP called SAP130, which is hepatocyte specific. So it's a DAMP that you really only see being secreted from dying hepatocytes. So we thought that Minkle might play this important role in hepatocyte cell clearance, as well as generally in the immune system for dealing with different fungal infections, as well as M. tuberculosis, as well as thought to bind other bacteria. It's really thought to do a lot of stuff. But what our collaborators found, Xiaoxia Li's group, which is down the hall from us, she found that under this low dose LPS, you get this rapid upregulation of Minkle. And it actually has a functional consequence too. So if you were to treat with a low dose LPS first, and then, so if you were to treat these cells, and in this case, these are bone marrow derived macrophages, if you were to treat them with SAP130 or TDB, you get a very mild response because Minkle is not upregulated yet. But if you were to pre-treat these cells with LPS, and I think they did this for 24 hours, you get this huge upregulation of Minkle, which leads to now the cells are responsive to TDB and SAP130, both by TNF and MCP1. So then what they did was they did a Minkle knockout in mice, and then they did our alcohol assay where we basically feed the mice alcohol chronically for, I want to say this was about 4 weeks. And then they looked at these liver enzymes, ALT and AST, and what we see is the Minkle knockout was in fact protective, and there are decreased triglycerides in the ethanol-fed mice. All right. So then, related to this, we see this other gene, dectin-1, as being important in alcohol liver disease. So this other group, Bernd Schnabel's group at UCSD, he found that during alcohol consumption, there's a huge increase in gut fungi, specifically Candida albicans. And the main receptor for Candida albicans is dectin-1, which senses, and it's also called CLEC7A, and it senses specific cell wall things in Candida albicans. And he also showed the same thing. In the same alcohol model, if you knock out CLEC7A, you get a decrease, you get rescue from the ethanol phenotypes. So decreased ALT, decreased triglycerides, and in fact, even decreased infiltrating cells into the liver. All right, so I'm interested in Minkle and these genes because of one very specific thing about them in the genome. And I would say my long-term research interests are weird stuff in the genome. So Minkle is present in what is called the NK receptor locus in the genome. This is on chromosome 12. And what's interesting is Minkle and two related genes, dectin-2 and dectin-3, sit really close to each other on the genome. A little farther away is this gene called DCIR, which is thought to sense malaria and viruses and all sorts of stuff. And then quite a bit farther away is actually dectin-1 nearby. So the question I had was, if Minkle is upregulated in low-dose LPS, and these genes are right next to each other, is there carryover upregulation of these genes? So what we can do is we can actually do this using single-cell data. And it's a little computationally difficult to explain how this was done. But the idea here was if we see upregulation of Minkle, which is also called CLEC4E, we should see upregulation of nearby genes. And so what I did was is I looked in all monocytes from each of my different conditions and asked, how often do you see correlated expression in single cells between one gene and every other gene in the genome? So what I did is I did this correlation for every gene and every other gene in the genome, and then just basically asked, how often do you see significance in different regions I'm interested in? And in the healthy control basal, you can see there's some correlation. The healthy control is a little weak in general, but once again, there is correlated expression between Minkle and the neighboring gene, CLEC. This is dectin-3, as well as, in fact, this non-coding RNA that's nearby. But when we go into AH, we see that this correlation expands quite significantly. And in fact, CLEC4E, CLEC4D, so this is Minkle, dectin-3, dectin-2, that non-coding RNA, they're all highly correlated within single cells. And then with LPS, actually, it seems like the correlation expands to include CLEC4A, which was DCIR, which is a little bit farther away. All right, and also just to add to this is a little bit unexpected, but dectin-1, CLEC7A is also nearby a few other genes, and those have highly correlated expression in AH. I also want to point out one other thing. This is not the only region. Actually, a lot of immune genes are clustered in the genome, and another example are in fact the chemokines. So CXCL81532, and this is just another gene nearby, sit right next to each other in the genome. And just looking at this kind of with your eyes crossed a little, they all look very similar. So I had the same question: does the chemokines have correlated expression? Because they sit next to each other in the genome, even though this genome region is quite a bit bigger than the Minkle region I was showing you before. And what we see is that is in fact true. So here's the chemokine cassette, and here are the CXCLs. In healthy control basal, where they're unstimulated, we see little correlation. But in response to LPS, we see that the correlation goes up significantly. Now that the genes are expressed, they're starting to have correlated expression. In AH, basally, we get some increase. It's already basally somewhat higher expressed in AH, and we see some correlated expression. And in fact, after LPS, we see a much stronger correlation expression. And the reason why this is shown like this is, one hypothesis I had was this could have been all an artifact due to how the correlations were measured. So the next question I had was, if this is in fact an artifact because of math, then I would expect that certain cells with more in-depth sequencing and that were normalized incorrectly, that these clusters would correlate with each other. And if that were the case, then I would be barking down like an artifact. But what I see though was in fact that these two clusters, while independently correlate with each other, they themselves don't correlate with each other, which is a good confirmation that something mathematical didn't go wrong. So I want to follow up on all of this going back into the liver, because the reason why we were looking at this to begin with in monocytes was because the idea is that these genes go into the liver, is that these cells infiltrate into the liver and exacerbate inflammation. So what we did was a paper came out a few years ago where they basically took, they did RNA-seq on a ton of different liver diseases. So they specifically did healthy control livers, early AH. So these are AH patients that aren't particularly sick, as well as severe AH. So, and they had two categories of AH based on how this was extracted. AHL are severe liver AH patients. XAH are patients that had AH, but had to undergo emergency liver biopsy because they were so sick. So you can think of this as being increasing degree of disease severity. And then NAFLD is our sister disease. Instead of alcohol-associated liver disease, this is non-alcohol-associated fatty liver disease, which in Europe, I'm pretty sure is being renamed metabolic-associated fatty liver disease, I think. Where the idea here is this is due to diet, poor diet, or some other metabolic factor, as well as this paper also looked at HCV and HCV cirrhosis. And what we observed from looking at their RNA-seq data was that these genes that we were interested in are in fact upregulated. So in AH, DCR1's upregulated. Dectin-2 in general is really low expressed, so we couldn't conclude anything from that. But Dectin-3 is upregulated, Minkle is upregulated, Dectin-1 was upregulated, and another gene which I didn't talk about very much, but MDL is also upregulated in AH. So that's basically all I want to tell you today. Here's basically what our model is, kind of combining everything. The idea here is that after gut disruption, we think that what's happening is there's a secondary immune surveillance program being activated. And how this works is in response to gut barrier dysfunction and low dose LPS, the immune system is highly geared towards sensing extraordinarily low concentrations of LPS, down to, there are reports that say that your immune system can detect 50, 10, even picograms per mil of LPS. So if your immune system is so sensitive to LPS, what happens then is it detects this low dose LPS, and some cells can respond in an inflammatory way, producing chemokines, but other cells diversify slightly and upregulate these C-type lectin genes, which all sit next to each other in the genome. And the idea here is it's to increase sensitivity for what other deleterious products may have come from the gut. So the LPS, in a sense, would be the first signal of saying there's something bad happening in the immune system. We don't know if there's fungi or viruses here, but why don't we upregulate the receptors temporarily to sense something else coming along? And then in that way we can detect whatever's going bad. And then in AH, because AH is this kind of chronic LPS stimulation, these are chronically upregulated, and the cells are much more prone towards pro-inflammatory gene expression. All right, and then the last thing I want to point out is this is all funded by K99 award I got in the US, where the idea was I would look at different aspects of this. The first aim of my grant was really to look at how the upregulation of these genes, we've now established that these genes are upregulated, but to what extent in kind of a biochemical way are they more sensitive to other byproducts of their targets? So for example, whatever binds Dectin-3, can these cells after low dose LPS now sense these targets better? The second goal was to do the single cell RNA-seq, as well as to look more specifically at Dectin-1 as kind of being an offshoot of the cluster, being a little bit farther away, having its own role in disease. And the final thing was really to look at in the genome, understand how this genome cluster is regulated using epigenetic techniques, chromatin capture, that kind of stuff. And so with that, I'd like to thank the lab, and I will take any questions.
Thanks so much for that, Adam. That was a really great talk. Really enjoyed that. So just to remind everyone, you can write your questions in the question and answer box, or you can raise your hand and I can unmute you. We've already got a question from Alba. So great talk, thank you. You mentioned a decrease in monocyte percentage in alcoholic hepatitis. Did you have a chance to look at absolute numbers and see if monocyte population was reduced?
So other people have done that, and it's actually not reduced. The monocytes are increased in AH. And in general, it's thought that leukocyte, what is the term? Increased leukocytes is a characteristic aspect of AH. But, people have done it in a couple different ways. There are definitely more leukocytes in general in AH. And from our proportions, we also see more monocytes as well. So both things are true. There are more immune cells in general in the blood, as well as there are more monocytes specifically.
That's really interesting. I was wondering sort of a bit of a broader question. So I studied non-alcoholic fatty liver disease for my PhD, but not the immune side of it at all. So once you move past steatosis and get to the like steatohepatitis or alcoholic hepatitis, is there a similar immune response? Do you know, or is that known?
Yeah, so it's actually super interesting. So in NAFLD, the equivalent is NASH, and there are a lot of similarities between NASH and ASH, but the major kind of health standpoint is NASH is not really severe. It doesn't really kill people, but NASH does increase fibrosis. So it's an interesting thing. If you were to have ASH, you will get severely sick and go to the hospital, and probably half of you would die. But with NASH, you may not ever go to the hospital for NASH. You may actually live a big portion of your life not knowing you have NASH. And as a result, the NASH itself increases the amount of fibrosis and increases the liver disease you receive. And it's thought that, and this is my hypothesis really, but the thought is that it's because while peripherally the immune response looks the same, there's increasing infiltrating monocytes and infiltrating neutrophils as well. It's thought that in ASH, they're highly pro-inflammatory, but in NASH, the immune cells don't do anything. They're kind of dysfunctional immune cells. They're unable to respond to infection as well. A lot of this work is done by this guy in Germany, Frank Tecki. He's looked at NASH using single cell data, and he sees that a lot of the immune function is lost in NASH.
That's really, really interesting. I'll probably have to pick your brains about that.
Yeah, of course.
At a later time, because we've got more questions coming in. So Eugenio is asking, does the constant leaking of LPS, could the constant leaking of LPS induce a trained immunity-like state? And I'm thinking particularly if changes could be caused by changes in the hepatic stellate cells or the myeloid progenitors.
Yeah, so we think of, so it's so complicated because, and you might know better than me, but LPS is thought to have two responses. There's, in an immediate LPS response, you get an immediate tolerance. So I think LPS downregulates TLR4 is kind of the mechanistic way of looking at that. And we actually do see that. We see that in our low dose LPS, TLR4 is downregulated. So on one hand, I think that is what you want to have happen. But then in AH, we, because of these other mechanisms, because of the pro-inflammatory nature, that doesn't happen as strongly. And for some reason, the immune tolerance that is supposed to happen in response to LPS just is immune exacerbation. Does that make sense? So we're not getting the proper signaling happening. And what ends up happening is that the LPS does not have a tolerant phase. But that's not totally true either, because there are other things where it does that are a little different. So it's complicated. And it's also complicated too, because the mouse models, which a lot of us use, it's also a little different, to be honest.
It's one of the great things about immunology, all of the exceptions to the rules.
Yeah, and it's so confusing sometimes. And what's even more confusing is the world of LPS is so concentration dependent and time frame dependent that you could look at two different papers that say two different things, and it's only different because the concentration was different, or one person looked at 1 hour and the other person looked at 10 hours,
Yeah, just to add fun to all of the experiments. So Ikram is saying, thank you so much for the presentation, and was wondering, is one of the parasites present on some of your slides leishmania?
I don't know. I think so.
And
so parasites are weird, right? Yeah, I don't think so, actually.
Yeah, I think one that would be interesting to study, I mean, this is completely off topic, but is it schistosoma, because it lays eggs in the liver? But that's probably a conversation for another day. And then Eugenio is also asking, has anyone looked at dysfunction of neutrophils in alcoholic ASH? Yep.
Yes, neutrophils are the #1 topic. If anything, I would say 50% of the immune people look at neutrophils, 50% look at monocytes. Neutrophils are harder to look at in a patient setting. We kind of, and I was thinking from kind of a technical standpoint, how would you even do this? But most people who do single cell RNA-seq and PBMCs regret not just doing it in whole blood. But I don't think you can just freeze whole blood for single cell RNA-seq. It would have to be directly from the patients. It would go straight into the sequencer. But that would be the only way to get neutrophils. I don't think there's any way you can kind of take whole blood, freeze it, and then unfreeze it, and then do the sequencing. From a clinical standpoint, especially here in the US, it would be technically very challenging to really look at the neutrophils from patients comprehensively and easily. I hear, though, that in Europe, it's like maybe a little bit easier to do some of that from patients. But
Do you think it might be possible then to do something like single nucleus sequencing of neutrophils? Because I was reading that you can get the nuclei from the frozen tissue and then
yeah.
do sequencing on that.
So that is a big push for kind of people who are nitpicky about extremely controlled experiments. And I'm kind of one of those people. I don't really like the idea of sequencing a different patient every single day whenever the sample comes in. So the idea is that, as the samples come in, you freeze them. And then in one day, you do this whole prep on a bunch. And I think nucleus is good. But aren't nuclei and neutrophils messed up?
Oh, that's true. That is,
So I actually don't know how that would look. And I actually don't know if anyone's done it comprehensively.
I guess if no one's done it yet, considering how long the technology's been around for now, it must be more complex than I'm thinking. Okay, well, if we don't have any more questions, I guess we can start to wrap this up. Would you mind just putting your contact details in the chat box, if that's okay, if people have any follow-up questions? And yeah, just while Adam does that, just to remind you, we have our last meeting next week at 4 p.m. GMT. And thank you again, Adam, for that talk. It was really, really great. Really enjoyed it. And thank you everybody for coming. And have a great week, everyone. And see you all next week.
All right. Thanks.
Thanks. Bye.